Hello everyone, myself Himanshu Samal. Today I going to be discuss one of my research topic that is curcumin in situ gel for the local treatment of periodontitis, its preparation, in vitro evaluation and its clinical assessment. As you know, periodontitis is an inflammatory disease of the gums which involves the degeneration of the periodontal ligament that leads to formation of a periodontal pocket. So there are several treatment for this periodontitis like scaling root planning, treatment of along with various antibiotic treatment. But there are several disadvantages or demerits associated with this conventional therapy, like insufficient drug concentration at the target site, chance of bacterial resistance, incapability to provide the sustained drug release. So, as this disease is confined to the periodontal pocket or periodontal cavity, local delivery of the drug in the pocket itself a best option for the treatment of periodontitis. Curcumin, a hydrophobic polyphenolic compound which shows anti-inflammatory, antioxidant, antimicrobial, antiseptic, anti-mutagen properties. Along with in addition this, it has pain suppression and wound healing capacity, and it is recognized as a non-toxic and safe material. That's why we are using, instead of this antibiotic treatment, we are using this curcumin for the treatment of periodontal disease. The basic objective of this study is to develop, characterize, and optimize curcumin-loaded dental gel to improve the bioavailability of the drug with a controlled release phenomenon for better therapeutic efficacy and increase the drug retention with an aim of improving the management of periodontitis. This is the basic plan of work, formulation, optimization, characterization of the gel formulation. After that, the optimized formulation is carried out for the clinical study, that is probing pocket depth, plaque index, sulcus breeding index. Next, come to the fabrication of curcumin gel. That curcumin gel is prepared by cold method by using thermosensitive polymer, that is Polaxmor 188 and 407. So first what we'll do, slow addition of the polymer, that is Polaxmor and methyl paraben in required quantity of cold deionized water. Then appropriate amount of curcumin is added to the above polymeric solution with continuous stirring. Then required quantity of carbopol and gel gum is added to the above drug polymer slurry. Finally, that slurry is kept in a refrigerator at 5 degrees Celsius in order to complete the dissolve the polymer solution, and then finally add small amount of polyethylene glycol. Next, after formulation, then this is the formulation table of the curcumin in-situ gel. We have to prepare 16 formulation. Then after formulation, all the formulation are carried out various physicochemical properties such as appearance, drug content, gelation temperature, viscosity, syringeability, gel strength, and pH. And if you see that particularly what we found that the prepared gel is found to be white transparent, and the percentage of drug, the pH of the preparation, pH of the gel formulation found to be 6.14 to 7.36. The gelation temperature found to be 30.2 to 36.5. The drug content is uniform in the gel formulation, which is found to be in the range of 98.9 to 100.2%. Then viscosity of the gel formulation were found to be 12,540 to 32,400 CPS at 25 degrees Celsius. Then the next part is we have to check the drug release. In vitro drug release study is carried out in flange diffusion cell using phosphate buffer. So what we have found that all the formulation, particularly CIG1 to C116, it shows initial bursting effect followed by control release. And if you see the drug release kinetic, what we found, the coefficient determination is highest in fast order. So it indicate all the formulation shows exhibit faster drug release kinetic. Also it is showing it is highest in Higuchi. So it will indicate the drug release mechanism is diffusion method. And if you see the Corsimer Papers model, found the Corsimer Papers release experiment, it is found it is equivalent to 0.5. That indicate it, and the drug release mechanism is Fickian. Then from all the above this, we can take that Polaxmor 407 and Carbopol P94 as the indifferent variable for the selected response, that is viscosity and gel temperature. So this is the optimization. We have taken Polaximor 407 and Curcumin 94. After doing the using centered cubic design, this is the formulation table. Then we have checked the response. What we have found, that particular the response one, that is viscosity, and response to swelling index, that with increase the concentration of Polaximor, all these two factor increases. So next we have to optimize. So this is the optimized formulation. Next, the optimized formulation we carried out for the clinical study, and we carried out the clinical study. We have divided the clinical study into two category of make it in two group. Group one, which treat only scaling root planning, while the group B, in that after scaling root planning, they have taken curcumin gel. These are the evaluation parameter, that clinical parameter: periodontal index, periodontal probing depth, and circular beating index. What we have found, as compared to SRP, the gel, the Curcumin In-Situ gel, shows effective for the treatment of periodontitis. This is the statistical analysis. This is the clinical application of the gel and dressing. So finally, the basic outcome of the research, we found that a curcumin, a well-known safety molecule with its multidisciplinary medicinal function, and that it is a well drug delivery system for the treatment of periodontitis. Thank you.